Welcome to this lecture on differentiation and growth in developmental biology. Have you ever wondered how a single microscopic egg transforms into a complex, multi-trillion cell human being? It is one of the most profound mysteries in all of science. Today, we are going to uncover the two fundamental processes that make this miraculous transformation possible. We will explore not just what these processes are, but how they interact, and even how we can use mathematics to predict and understand them. Let us dive in. To understand our topic, imagine you are tasked with building a massive, complex city, but you only have a single brick to start with. First, you need to manufacture millions of identical bricks just to have enough raw material. In biology, this rapid multiplication of raw material is what we call growth. But a city made entirely of identical bricks is just a giant, useless pile of clay. To make buildings, bridges, and windows, you need to transform some of those materials into steel, wood, and glass. This transformation into specialized materials is exactly what differentiation is. You need both to build a functioning system. Now that we have our analogy, let us look at the formal biological definitions. In developmental biology, growth is defined as the irreversible increase in the physical size and mass of an organism. How does an embryo achieve this? It happens primarily through a process of cell division, known as mitosis, where one cell splits into two, two into four, and so on. Additionally, cells secrete structural proteins and fluids, known as the extracellular matrix, which also adds to the organism's mass. Ultimately, the primary purpose of growth is to provide the massive amount of raw cellular material required to construct the body. While growth gives us the material, differentiation gives that material a purpose. Differentiation is the complex cellular process where an unspecialized, generic stem cell transforms into a highly specialized cell type. This is not a temporary change. It involves permanent alterations in gene expression. Certain genes are turned on and others are turned off. Because of this, a generic embryonic cell can become a beating heart muscle cell, a thinking brain neuron, or a protective skin cell. Each specialized cell develops a distinct structure, perfectly suited to its specific function within the body. These two processes do not happen in isolation. They are tightly coupled and highly regulated by complex genetic networks and chemical signals. Interestingly, there is often a biological trade-off between the two. A cell must frequently exit the rapid growth phase of the cell cycle in order to fully differentiate into its final mature form. It stops dividing so it can specialize. Working together in perfect harmony, growth and differentiation drive a process called morphogenesis. Morphogenesis is the creation of the organism's overall shape, structural organization, and functional anatomy. Why is it so critical for us to understand this delicate balance? In the real world, this knowledge forms the entire foundation of regenerative medicine. Scientists are learning to coax stem cells to differentiate into replacement tissues, like growing new skin for burn victims. Furthermore, when the balance between growth and differentiation breaks down, the results can be devastating. Cancer is fundamentally a disease where cells grow uncontrollably and fail to differentiate into useful tissue. By studying these developmental processes, medical researchers can develop targeted therapies to force cancer cells to stop dividing or to repair congenital birth defects. Now that we understand the biology, let us look at the mathematics of early development. To understand how an embryo rapidly increases its cell number, We use the exponential growth model. Conceptually, this applies when the rate of growth is directly proportional to the current number of cells. The more cells you have, the faster the population grows. In this formula, n of t represents the total number of cells at a given time t. N naught is the initial number of cells we start with. The constant e is the base of the natural logarithm. The variable r is the intrinsic growth rate. If the growth rate r increases, the cell population explodes much faster. If r decreases, population growth slows down, which often happens when cells begin to differentiate. Let us apply this formula with a concrete example. During the early cleavage stage of a frog embryo, a single fertilized egg divides rapidly. We are given an initial cell count of 1, a continuous growth rate of 0.693 per hour, and a time of 10 hours. We want to find the final number of cells. We chose the exponential model because early embryonic cleavage involves rapid, continuous doubling. We substitute our values into the equation, multiplying the rate and time in the exponent to get 6.93. Evaluating e to the power of 6.93 gives us our final answer. The embryo will have exactly 1,024 cells. Physically, this represents 10 perfect rounds of cellular doubling, providing massive raw material for the embryo. Our previous example assumed the whole embryo grows uniformly, but biology is rarely that simple. As an organism develops, different parts of its body often grow at completely different rates. This concept is known as allometric growth. Think about a human baby. Its head is proportionally much larger than an adult's head. This differential growth is deeply tied to differentiation. As tissues specialize to perform unique functions, they must develop distinct sizes and proportions relative to the whole body. Allometric growth explains how localized growth and differentiation work together to create the unique, changing proportions of a developing embryo. To track these changing proportions, we use the allometric growth equation. Conceptually, this formula compares the growth of a specific part to the growth of the whole. In this equation, the variable y represents the size or mass of a specific organ, like the brain or a limb. The variable x represents the size or mass of the entire organism. The symbol b is a constant that relates their initial sizes at the start of development. The most critical part of this formula is the exponent a, known as the allometric growth ratio. If the exponent a increases, it means the specific organ is growing at a much faster rate compared to the rest of the body. Let us look closer at that allometric growth ratio, the exponent a, because it tells us exactly what is happening physically. If a is exactly equal to 1, the organ grows at the exact same rate as the overall body. We call this isometric growth, meaning equal measure. However, if a is greater than 1, the organ is growing faster than the rest of the body, taking up a larger proportion over time. Conversely, if a is less than 1, the organ grows slower than the body. As the organism gets larger, that specific organ will become proportionally smaller. This simple exponent perfectly captures the changing geometry of life. Let us see this in action with human fetal development. We are given an allometric equation where head mass, y, equals 0.5 times total body mass, x, to the power of 0.8. We need to find the increase in head mass as the fetus grows from 1,000 grams to 3,000 grams. We use the allometric formula because the head and body grow at different rates. First, we plug in the initial body mass of 1,000 to find the starting head mass of 125.6 grams.
Next, we plug in the final body mass of 3,000 to get 302.2 grams. Subtracting the two gives our final answer. The head mass increases by 176.6 grams. Because the exponent 0.8 is less than 1, the head is growing slower than the overall body.
Now that we have calculated these changes, let us visualize the relationship between our two main concepts. Look at this graph. The horizontal axis represents developmental time, from early to late. The vertical axis represents the relative rate of cellular activity. Notice the two curves. The curve for cell proliferation, or growth, starts very high early in development, but steadily declines. In contrast, the curve for cell differentiation starts low and rises dramatically over time. The most important feature is where they cross. This intersection represents a fundamental biological trade-off. It visually proves what we discussed earlier. As development progresses, cells must literally slow down their rapid division in order to specialize and take on their final functional roles. We have covered a lot of ground today, so let us summarize the most important ideas. First, growth and differentiation are the two intertwined pillars of developmental biology. Growth provides the necessary cellular mass through rapid division, while differentiation assigns specific, specialized roles to those cells so they can actually function. Second, we can track these complex biological processes using mathematics. Exponential models help us understand rapid early cell proliferation, while allometric models allow us to track how different tissues develop proportionally. Finally, remember that the delicate balance between multiplying and specializing is the absolute key to grasping how a single microscopic egg successfully builds a fully functional, complex living organism. Thank you for watching.